Hello everyone and Assalamualaikum. The topic for this video is glomerular and tubular functions. There are 3 learning outcomes for this lecture. So by the end of this lecture, you should be able to describe the mechanism of tubular reabsorption and secretions, transport maximum and renal threshold, as well as water and sodium homeostasis. There are basically 3 basic processes of urine formation. First is glomerular filtration, where the blood in the glomerular capillaries is filtered through the filtration membrane. So filtrates from glomerulus will enter the proximal tubule, whereas unfiltered substances will flow to the efferent arteriole and to the peritubular capillary. Second process is reabsorption. Water and solutes such as sodium, chloride, bicarbonate, glucose, amino acids, and urea are removed from the tubular lumen and transported back into the peritubular capillaries. So reabsorption process occurs throughout the renal tubule. Depends on several factors such as the type of substances, availability of transporter, as well as availability of hormone. The third process is secretion, which is transport of unwanted substances such as organic acids, organic bases, and potassium from the tubular capillaries back into the tubular lumen. So similar with reabsorption process, the secretion mechanism also involves transporters. The next process is excretion process, which is the elimination of urine from the collecting duct to the ureter via renal pelvis. Glomerular filtration has been covered in the previous physiology lecture, Introduction to Renal Function and Clearance Concept. So for this lecture, we are going to look at reabsorption process and secretion. Excretion or micturition will be taught in the next lecture, urine concentration mechanism and micturition. There are several mechanisms of tubular reabsorption. The 2 basic transport mechanisms are via paracellular pathway and transcellular pathway. Paracellular pathway is when the filtrates or substances in the lumen pass through the epithelial layer of the tubule in between the tubular cells. This is paracellular pathway, in between 2 cells. For transcellular pathway, the way the substances reabsorb from the lumen into the peritubular capillary is through the tubular cells. So these are the pathways that substances can get through from lumen into the tubular capillary, either paracellular pathway or transcellular pathway. So the mechanism by which this transportation occurs depends on the type of substances, transporters, and the need for energy. The transport mechanism that does not need the use of energy are diffusion, which can be further categorized into simple and facilitated diffusion. Both simple and facilitated diffusion are passive process that does not require energy. Solutes or molecules move from higher concentration gradient to the lower concentration gradient. So in the reabsorption process in the tubule, if the concentration of substances is higher in the lumen, the substances will diffuse from the lumen into the tubular capillary, either via paracellular pathway or transcellular pathway. Facilitated diffusion depends on transporters or carrier proteins located on the tubular cells. On the other hand, water is transported via paracellular and transcellular pathway via osmosis. Tubular reabsorption may also occur via active process, which are primary and secondary active transport. In active transport, the concentration of the substance may be higher in the tubular cells or capillary than in the lumen. So reabsorption will require energy because the substance goes against its concentration gradient. In primary active transport, the energy comes from ATP. For example, sodium-potassium ATPase pump. In secondary active transport, the energy comes from the primary active transport. There are 2 types of secondary active transport, which are co-transport and counter-transport. For co-transport, the two molecules are translocated into the same direction, whereas for counter-transport, the two molecules are translocated across the opposite direction. Now let us take a look into the tubular reabsorption process, tubules by tubules. We need to understand that each of the renal tubule have their own unique characteristics. Some part may have certain type of transporters, and some do not have those transporters. So some part of renal tubules may reabsorb certain substances while others do not. Proximal tubule reabsorb about 65% of the filtered water, sodium, chloride, and potassium. It also reabsorbs about 90% of filtered bicarbonate. Proximal tubule also absorb almost all of the glucose and amino acids from the filtrates, which means when the filtrates leave the proximal tubule, it usually does not contain any glucose or amino acids. There are presence of extensive brush border on the epithelial surface of the luminal membrane, so this will increase the surface area for absorption. There are also some secretion process going on in the proximal tubule. Secretion means the substance moves from peritubular capillary into the lumen, opposed to the reabsorption. So proximal tubules secrete excess hydrogen ions, organic acids, and bases. Reabsorption process in the proximal tubule occur mainly via active process, either primary or secondary active transport. Reabsorption process is quite different in the early and late proximal tubule. So this is the cell of proximal tubule. This is the lumen, meaning that this is the luminal membrane. And this is the blood in the peritubular capillary, meaning this is basolateral membrane. So in the early proximal tubule, sodium is reabsorbed from the lumen into the cells via co-transport, such as sodium glucose co-transporter. This is also called SGLT, which stands for sodium dependent glucose co-transporter, SGLT. There are also other co-transporters such as sodium amino acid co-transporter, sodium phosphate co-transporter, sodium lactate or sodium citrate co-transporter. And then sodium is extruded from the cell into the blood via sodium-potassium ATPase located at the basolateral membrane. Glucose and other solutes are extruded into the blood via facilitated diffusion. There is one counter-transport in the luminal membrane of the early proximal tubule, which are sodium-hydrogen exchanger. Sodium is reabsorbed into the cell in exchange for hydrogen. So this will cause secretion of bicarbonate from the cell into the blood. When the filtrate reach the late proximal tubule, all the filtered glucose and amino acids have been reabsorbed into the blood. So the tubular fluid leaving the early proximal tubule contains a high level of chloride. So the late proximal tubule reabsorb mainly sodium and chloride. Sodium is reabsorbed via sodium-hydrogen exchanger at the luminal membrane and into the blood via sodium-potassium ATPase. Chloride is reabsorbed into the cell via chloride formate counter-transporter and into the blood via diffusion. Sodium and chloride are also transported via paracellular pathways. Let's take a look closer at glucose reabsorption. Glucose reabsorption mainly occurs in the early proximal tubule. When filtrate reach the end of proximal tubule, all of the glucose has been reabsorbed. As we have learned previously, the luminal membrane of the proximal tubule contains SGLT, sodium glucose co-transporter. So here, glucose is transported from the lumen into the cells via SGLT. This process is secondary active transport. The energy for this transport comes from sodium-potassium ATPase pump. At the peritubular or basolateral membrane, glucose is transported from the cell into the capillary blood by facilitated diffusion by protein carriers glucose transporter 2, GLUT2. The reabsorption of potassium at the proximal tubule is mostly via passive diffusion. There are also sodium-potassium ATPase pump that pump sodium out of the cells and potassium into the cells. But then, the higher level of potassium in the cells will cause potassium to diffuse out of the cells, either into the lumen or into the interstitium or blood. For bicarbonate, about 90% of bicarbonate is reabsorbed in the proximal tubule. The other 10% is reabsorbed in distal tubule and collecting duct. Bicarbonate is reabsorbed via passive diffusion. However, bicarbonate ion cannot cross the luminal membrane because it is negatively charged. So, the bicarbonate will combine with hydrogen ion to form carbonic acid. The carbonic acid will be dissociated into carbon dioxide and water by carbonic anhydrase. Carbon dioxide is lipid soluble, so it can easily enter proximal tubular cell. In the tubular cell, the opposite process occurs, whereby carbonic anhydrase will combine carbon dioxide with water and form carbonic acid. Carbonic acid will dissociate to form hydrogen ion and bicarbonate ion. On the basolateral membrane, there are sodium bicarbonate co-transporter and bicarbonate chloride countertransporter. So bicarbonate can be reabsorbed into the blood via these transporters. Reabsorption of water. Proximal tubule is highly permeable to water, so it does not need any hormone such as ADH to alter its permeability to water. Water will be reabsorbed into the capillary blood because of higher osmolarity or higher osmotic pressure in the capillary. This higher osmolarity in the blood is contributed by solute reabsorption that occurred earlier. Alright, let's take a look at this figure. This is the lumen and this is the blood in the peritubular capillary. So as we have learned earlier, at early proximal tubule, sodium reabsorption occurs. So the positive charge in the blood will attract negatively charged ions such as phosphate and bicarbonate to also enter the blood. So this will cause increase in osmotic pressure in the blood. So because of this higher osmotic pressure in the blood, water will be attracted to enter the blood. So water are reabsorbed from the lumen into the blood via osmosis. This process is not regulated by ADH. So those are the reabsorption process that occur in the proximal tubule. At loop of Henle, different reabsorption process occur. Loop of Henle consists of 3 limbs: thin descending, thin ascending, and thick ascending limb. And all of them have different reabsorptive characteristics. Thin descending limb is highly permeable to water, so it allows water reabsorption via aquaporin 1 water channels. About 20% of filtered water will be reabsorbed at the thin descending limb. Thin descending limb is also moderately permeable to solutes including urea and sodium. However, it does not reabsorb significant amount of solutes because this limb does not have any transporter or carrier proteins. So the solutes will be reabsorbed via simple diffusion. The permeability of the thin descending limb to urea is important for urea recycling process. On the other hand, the thin ascending limb, unlike the thin descending limb, it is not permeable to water, meaning water is not reabsorbed at this limb. This impermeability of the ascending limb to water is important for countercurrent multiplication process. This limb is permeable to urea. This permeability to urea is also important for urea recycling process. We will look into these 2 processes in the next lecture. Similar to the thin descending limb, the thin ascending limb also do not have transporter, so it does not reabsorb significant amount of solutes. Its reabsorptive capacity is much lower than the thick ascending limb. Similar to the thin ascending limb, the thick ascending limb is also impermeable to water. However, the thick ascending limb reabsorbs a significant amount of solutes because it has many types of transporters. One of the transporters is sodium potassium 2 chloride cotransporter, NKCC2. NKCC1 located throughout the body, but NKCC2 is located specifically in the kidney. This NKCC2 allow reabsorption of sodium, potassium, and chloride into the tubular cells. Higher potassium in the tubular cells will cause backleak of potassium back into the lumen, causing tubular fluid to be positively charged. This positive charge of tubular lumen will force or attract cations such as magnesium and calcium to diffuse into the interstitial fluid via paracellular diffusion. Early distal tubule has the same reabsorptive characteristics like the thick ascending limb, meaning it does not reabsorb water, but it reabsorbs most of sodium, potassium chloride, calcium as well as magnesium, whereby sodium chloride is reabsorbed via sodium chloride cotransporter. Both late distal tubule and collecting duct have similar functional characteristics. There are two cell types on the tubular lining, which are principal cells and intercalated cells. These two cell types reabsorb and secrete different solids. Principal cells reabsorb sodium and secrete potassium, whereas intercalated cells reabsorb potassium and bicarbonate and secrete hydrogen. At the basolateral membrane of principal cells, there is sodium-potassium ATPase pump that pumps sodium out of the cells and potassium into the cell. So this will cause sodium concentration inside the cells lower than the tubular lumen. So this will cause sodium to diffuse into the cells via epithelial sodium channel, ENAC. This process is governed by aldosterone. Presence of aldosterone will stimulate reabsorption of sodium by the principal cell. Intercalated cell, the basolateral membrane contains bicarbonate chloride countertransporter. So the function of this intercalated cell is to reabsorb bicarbonate. On the luminal membrane, there is hydrogen ATPase pump that actively secreted hydrogen from the cell into the tubular lumen. So aldosterone plays an important role in the reabsorption and secretion process in the late distal tubule and collecting duct. There are two important functions of aldosterone. In the principal cell, it increases the activity of sodium-potassium ATPase pump, which will cause sodium to be reabsorbed and potassium to be secreted. In the intercalated cells, aldosterone increases hydrogen ATPase activity, which will cause hydrogen secretion into the tubular lumen. With regards to water reabsorption of these two tubules, both late distal tubule and collecting duct are not permeable to water. However, in the presence of ADH or vasopressin, the permeability of these two tubules to water will increase. ADH, vasopressin, binds to vasopressin receptor and activates CAMP, and this will cause insertion of aquaporin or water channel into the apical membrane. So this will allow water to be reabsorbed into the blood via osmosis. This figure illustrates the importance of ADH in urine production. In the first situation, which is a hypothetical situation that our body does not produce ADH, or in the case of diabetes insipidus, the tubules that are permeable to water without ADH are proximal tubule as well as thin descending limb. So here water will be reabsorbed. At the thick ascending limb, a significant amount of solids are reabsorbed. So this will cause the luminal fluid that pass through the distal tubule and collecting duct to be hypoosmolar or diluted. So the urine that will produce not only diluted but also in large quantity, so large volume of diluted urine. But in the presence of ADH, ADH will increase permeability of distal tubule and collecting duct to water. So here water is reabsorbed. And the results is small volume of concentrated urine. So this is the importance of ADH to allow water reabsorption in the distal tubule and collecting duct. Now let's take a look at renal handling of sodium and water. First we look at sodium. At proximal tubule, about 60 to 70% of sodium are reabsorbed via either sodium hydrogen exchanger or sodium co-transporter. At the lobe of Henle, another 25 to 40% are reabsorbed via NKCC2. At the early distal tubule, another 5 to 8% of sodium are reabsorbed via sodium chloride co-transporter. And finally, at the late distal tubule and collecting duct, the remaining 2 to 3% of sodium are reabsorbed via sodium channels. Sodium reabsorption at the whole tubule require primary active transport of sodium-potassium ATPase located at the basolateral membrane in order to transport the sodium from the cell into the ICF or ISF or into the blood. Renal handling of water. About 65% of water are reabsorbed at the proximal tubule via osmosis. Proximal tubule is permeable to water, so reabsorption of water does not require any hormones. At the lobe of Henle, only thin descending limb is permeable to water. So only about 10% of water are reabsorbed. There is no water reabsorption at the early distal tubule, whereas at the late distal tubule and collecting duct, it depends on the body hydration state. During water loading, or when we just drink a large amount of water, only about 3% of water are reabsorbed. Whereas during dehydration, more than 24% are reabsorbed. So here at the late distal tubule and collecting duct, osmoreceptor plays the important role to detect the state of hydration of our body, or osmolarity, and then to release appropriate amount of ADH. So appropriate amount of water are reabsorbed. We will look at osmoreceptor ADH system shortly in the next few slides. So these are the summary of renal handling of sodium and water. A little bit on tubular secretion. Tubular secretion is the reverse process of tubular reabsorption. Substances are transported from the blood or peritubular capillary into tubular cells and into the tubular lumen. The substances include potassium, hydrogen, ammonium, creatinine, urea, and some hormones and drugs such as penicillin. Secretion process is important for eliminating substances that were not filtered, eliminating substances that were passively reabsorbed but need to be removed, to control blood pH, and to eliminate excess potassium in the body. Now we are moving to the second learning outcome for this topic regarding glucose reabsorption, which are transport maximum and renal threshold. Transport maximum. It is the maximum rate at which a substance can be reabsorbed from the renal tubule, or a filtered substance exceeds the capacity of the carrier proteins and specific enzyme involved in the transport process. So, what does that mean? Okay, in order to understand this, let's take one substance as an example, glucose. So normally, all of the glucose that are filtered will be reabsorbed, particularly at the early proximal tubule. Glucose reabsorption requires SGLT2 as the transporter, but the amount of SGLT2 is limited. So Tm is when all of this SGLT2 are occupied, so glucose cannot be absorbed anymore. This will cause glucose to appear in the urine. So Tm is the reabsorption rate at which all nephrons have reached their maximal capacity to reabsorb glucose. Tm for glucose is 375 mg per minute in men and 300 mg per minute in women. Now, renal threshold. Renal threshold is the plasma level of glucose at which glucose starts appearing in the urine. Let's take a look at this graph. This is glucose filtered load, the red line. Increase in plasma glucose concentration resulted increase in filtered load, meaning amount of glucose in plasma is proportional to the glucose amount being filtered. The blue line represents reabsorption. Increase in glucose concentration resulted increase in reabsorption. But when transport maximum is reached, which is about 375 mg per minute in men and 300 mg per minute in women, reabsorption process will stop because all SGLT2 are occupied with glucose. The green line represents glucose excretion. If plasma glucose concentration reaches 200 mg per 100 ml of plasma, this means that renal threshold has been reached. So therefore, glucose will start to appear in the urine. And if plasma glucose concentration keeps on rising, glucose excretion rate will also increase. If you notice, glucose starts to appear in the urine even before transport maximum is reached. Here's why. This is because not all nephrons have the same TM for glucose. Some nephrons may have more transporters than the others. Like bigger nephrons like juxtamedullary nephrons may have more transporters compared to the cortical nephrons. So, the bigger nephrons may have higher TM value. So the smaller nephrons may excrete glucose before other nephrons reach their TM. The overall TM for the kidneys is when all nephrons have reached their TM. Osmotic diuresis is increased urination, which is caused by the presence of certain substances that cannot be reabsorbed due to either pathological state or the normal nature of the substance. For example, when filtered glucose is more than the TM value, glucose can no longer be reabsorbed, so it will stay in the lumen. The presence of glucose in the lumen will increase the osmotic pressure in the lumen, and this will cause water retention in the lumen in order to balance out the osmotic pressure. So this water retention in the lumen will then cause urine output to increase, meaning there will be large volume of urine. So increase in urine volume is termed diuresis. So this is osmotic diuresis. The third learning outcome for this lecture is water and sodium homeostasis. So basically, water and sodium homeostasis is how the volume of water or the amount of sodium are balanced in our body. Body fluid osmolarity is maintained at a value of about 290 milliosmol per liter, or for simplicity, 300 milliosmol per liter. Even small deviations in body fluid osmolarity will produce hormonal responses that will alter water reabsorption by the kidneys, which will attempt to return osmolarity back toward the normal value. Variations in water reabsorption produce variations in urine osmolarity. Urine osmolarity can vary from as low as 50 milliosmol per liter to as high as 1,200 milliosmol per liter. When urine osmolarity is equal to blood osmolarity, it is called isoosmotic urine. When urine osmolarity is higher than blood osmolarity, it is called hyperosmotic urine, whereas when urine osmolarity is lower than in the blood, it is called hypoosmotic urine. Sodium is the major cations in ECF. Sodium plasma concentration is normally regulated within close range of 140 to 145 milliequivalent per liter. Sodium homeostasis is important because sodium concentration in the ECF will determine ECF volume, blood volume, and blood pressure. For example, if there is an increase in the amount of sodium in the body, this will lead ECF volume, blood volume, and blood pressure to also increase. And vice versa, if sodium concentration in the plasma is decreased, this will cause ECF volume, blood volume, and blood pressure to decrease. Kidney is the major regulator of water and sodium homeostasis. However, it is important to note that kidney can only minimize fluid loss. Intake is required in order to add fluids in the body. There are basically three mechanisms for water and sodium homeostasis. osmoreceptor ADH system, thirst mechanism, and renin-angiotensin-aldosterone system. Osmoreceptor ADH system. This figure shows the basic components of the osmoreceptor ADH feedback system for control of ECF sodium concentration and osmolarity. And this figure shows the neuroanatomy of the hypothalamus and the pituitary gland, where ADH is synthesized and released. The hypothalamus contains two types of neurons that synthesize ADH, which are supraoptic neuron and paraventricular neuron, where both of these neurons have axonal extensions to the posterior pituitary. Once ADH is synthesized, it is transported down the axons of the neurons into the posterior pituitary gland, and it will be stored in vesicles. When ECF osmolarity is increased above normal, or plasma sodium concentration increase, for example in the case of water deficit, osmoreceptor cells, which is a special nerve cell located in the anterior hypothalamus, will shrink. This shrinkage will cause them to send signal to the supraoptic nuclei, which then relay the signals down the stalk of the pituitary gland to the posterior pituitary. This action potential will stimulate the release of ADH. ADH will enter the bloodstream and is transported to the kidneys, where it increases the water permeability of the late distal tubule and collecting duct. And this will cause increased water reabsorption, and as a result, small volume of concentrated urine will be produced. So therefore, water is conserved in the body, while sodium and other solids continue to be excreted in the urine. And this causes dilution of the solids in the ECF, and thereby correcting the initial concentrated ECF. The opposite sequence of events occur when the ECF becomes too diluted or hypoosmotic. For example, with excess water ingestion, ADH secretion will decrease. The permeability of renal tubule to water will also decrease, so less water will be reabsorbed. So the result is large volume of diluted urine. So this in turn concentrates the body fluids and return plasma osmolarity towards normal. The osmoreceptor ADH system work together with the thirst mechanism to regulate ECF osmolarity and sodium concentration. Thirst is a sensation of dryness in the mouth and throat related to a need or desire to drink. When sodium concentration increases only about 2 milliequivalent per liter above normal, the thirst mechanism is activated, causing a desire to drink water. This is called threshold for drinking. There are several factors that may increase or decrease the sensation of thirst. Factors that may increase thirst include increased plasma osmolarity, decreased blood volume, decreased blood pressure, increase of angiotensin II, and dryness of mouth, whereas the factors that may decrease thirst include decreased plasma osmolarity, increased blood volume, increased blood pressure, decrease in angiotensin II, as well as gastric distension. Renin-angiotensin-aldosterone system is the most important hormonal system that regulates water balance and blood pressure. Renin is secreted by juxtaglomerular cells. Renin is released when there is either decrease in plasma osmolarity, ECF volume, or decrease in blood pressure or increase in sympathetic activity. Renin's function is to convert angiotensinogen that is produced by the liver to angiotensin I, whereas angiotensin I is converted to angiotensin II by angiotensin converting enzyme that is secreted by pulmonary and renal endothelium. Angiotensin II will stimulate sympathetic activity, stimulate reabsorption of sodium chloride, and excretion of potassium by the renal tubule. It also stimulates the adrenal gland to secrete aldosterone, whereby this aldosterone will also stimulate reabsorption of sodium chloride and excretion of potassium in the principal cell of late distal tubule and collecting duct. Angiotensin II will also stimulate arteriolar vasoconstriction, causing increase in blood pressure. Aside from that, angiotensin II will stimulate the pituitary gland to secrete ADH, which will increase the permeability of the late distal tubule and collecting duct to water, hence will increase water reabsorption. So the role of angiotensin II ultimately leads to increased sodium and water reabsorption. Opposite action of angiotensin II, there is also hormone that leads to increased sodium and water excretion. One of these hormones is atrial natriuretic peptide, or ANP. It is released by cardiac atrial muscle fibers. Elevated levels of ANP are found during hypervolumic state, or increase in blood volume. Increase in blood volume will cause atrial myocardial cells to stretch, which will cause ANP to be released. ANP will inhibit the hypothalamus to release ADH. It will also increase GFR while decreasing renal secretion in the kidney. ANP will inhibit aldosterone secretion by the adrenal cortex. It also signals the medulla oblongata to decrease blood pressure. So all of this ANP action will lead to increase in sodium and water excretion in the urine. Effective circulating volume, or ECV, refers to the volume of ECF that is in the arterial system and is effectively perfusing the tissues. The blood volume, blood pressure, and cardiac output are dependent on ECV. The regulation of sodium balance is closely related to the maintenance of ECV. A low ECV will trigger 4 parallel effector pathways that act on the kidney. Each system consists of stimulus or signal, which is the ECV, sensors, which are volume or pressure sensors, effector, which is the kidney, and response, which is the alterations in sodium chloride excretion. The effect is either a change in hemodynamic or change in sodium transport by renal tubular cells. For example, in the case of decreased ECV, Renin-angiotensin-aldosterone system will be activated. Sympathetic activity will also be activated, and ADH secretion will be increased. At the same time, ANP secretion will be inhibited. So all of these effector systems will change the hemodynamic and tubular transport in the kidney, which ultimately leads to reduce in sodium excretion. So that's all for this topic, glomerular and tubular functions, that covers the mechanism of tubular reabsorption and secretion, transport maximum, renal threshold, as well as water and sodium homeostasis. If you have any question regarding this topic, please email me at kairno@usm.my. Thank you.